First, I would like to thank the organizers for giving me the opportunity to describe my work. And I will talk about identifying a landscape of DNA methylation-driven genes in cancer using a method that I developed called METHYLmix. First, a short introduction about DNA methylation. DNA methylation is the transfer of a methyl group to a CG dinucleotide, which is often called a CpG site. And this is known to cause gene expression silencing. And this process is often deregulated in cancer, causing either hypermethylation, where there's more methylation of a gene compared to normal, or hypomethylation, where there's less methylation in a particular cell compared to normal. Currently, there is no method yet to model hyper and hypomethylated genes in cancer. Also, another issue here is that the normal state for DNA methylation data is often unknown compared to, for example, copy number data, where we know we have 2 copies of a gene. And also, I want to focus on DNA methylation events that are functional. I want to focus on DNA methylation, cis-regulated DNA methylation that has an effect on gene expression. So I developed a new algorithm that is called METHYLmix to identify significantly hyper or hypomethylated genes in cancer. We did this by modeling these genes as a mixture of Gaussians. And essentially what we're doing is we're trying to identify subpopulations of samples that have similar methylation patterns. Next, we're going to compare these methylation states with normal DNA methylation levels. And finally, I'm also going to filter these genes and look if they can also explain gene expression data. So this is a typical methylation profile of a gene. So 1 means that the gene is fully methylated, whereas 0 means that the gene is unmethylated. And so this is a typical DNA methylation distribution. And so the algorithm that we developed is modeling this distribution as a mixture of Gaussians. And we have an iterative model building algorithm that will add mixture components to the model based on the Bayesian information criterion score. And we also use a cross-validation strategy to do this. So suppose for this distribution that 2 mixture components have the best BIC score. Next, so we have identified 2 methylation states for this gene, a low methylation state and a high methylation state. But we don't know what the normal state is, so now we are going to also map the normal state for this gene. Suppose that this is the normal state of this gene, so now we know that this methylation state is the normal situation for this gene, and this is a hypermethylation. And this gene is actually the BRCA1 gene in ovarian cancer based on the TCGA data analysis. So we have identified a formal way of identifying hypermethylation of BRCA1 in ovarian cancer. Finally, we also integrate paired gene expression data. So we also look at if the methylation levels are correlated with gene expression. And we do this with a linear model, and we use the R-square statistic to quantify the amount of variation that is explained by the methylation for the gene expression status of that gene. Next, we applied this method on 3 cancer data sets from the TCGA project. So we used the breast cancer, ovarian cancer, and the GBM data set. And we had both cancer, we used the cancer methylation data, the normal methylation data, and then paired gene expression data. And here you can see also the number of genes that we had for each of these cancer data sets. It's also important that the number of normal samples for the methylation data varied a lot. So we only had 4 samples for the GBM, where we had 27, for example, for the breast cancer data set. So when we applied our algorithm to the GBM data sets, so we start from 17,000 genes. Next, we found that 3,600 genes were differential, so they had a methylation state that was different from normal. Next, we found 743 genes where the methylation state was predictive of gene expression. And finally, we had 331 genes that were both differential and functional. So next, this is a plot where I show the comparison of all the 3 different cancer sites. So these are the numbers of hyper and hypomethylated genes that were identified by METHYL-NICs in each of these 3 cancers. Interestingly, there were also 13 genes that were common, hypermethylated in all these 3 cancers, and 2 genes were hypomethylated in all 3 cancers. For the hypermethylated genes, SIDAP is a well-known tumor suppressor gene that is often methylated in cancer. We also looked at which pathways that were enriched in all these cancers, because we don't expect that the same genes will be hyper or hypomethylated in each cancer. And we found that the hypermethylated genes were enriched in genes that are driving the differentiation from mesenchymal to epithelial state. And we also found genes that were upregulated after the knockdown of the embryonic stem cell machinery. So this suggests that cancer cells are in a stem cell-like state, and that genes that would drive away from this state are hypermethylated. Next, we looked at the properties of the CpG sites from these genes that were identified by METHYL-MIX. First, we looked at where the CpG sites were, and we looked if they were closer than 500 base pairs to the transcription start site versus further away. And we found that the genes that were functional, so the genes that are correlated to gene expression, that a higher proportion of these were closer to the transcription start site. For example, here in breast cancer. And we found that this was also true in ovarian cancer and GBM. It's also important to say that the number of genes that are correlated with gene expression is much lower than the total number of genes here for each of these cancers. Next, we also looked at where these CpG sites were with respect to CpG islands. So just a short introduction about CpG islands. Lots of CpG sites are clustered in the genome, and these clusters are called CpG islands, whereas the CpG sites that are close to CpG islands are called CpG shores, and all other CpG sites are just anywhere else. So we looked at where these hypermethylated and hypomethylated genes were for these 3 cancers, and for the hypermethylated genes, we found that they were enriched in the island, so they were always proportional to the islands. Whereas for the hypomethylated genes, they were enriched outside of CpG islands. So in all these 3 cancers, we found a higher proportion of hypomethylated genes outside of CpG islands compared to the platform distribution. And this is consistent with other reports. Next, we also defined a differential methylation value, or what's called what we call a DM value. And this value captures the differential methylation of each gene. So this is defined as the difference of each methylation state that we found by the mixture model versus the normal. For example, here for this hypermethylation state, we have a DM value which is bigger than 0. Whereas for this hypomethylation state, we have a DM value which is smaller than 0. And we use these DM values to cluster the samples and the data to see if we can find known or new subgroups. For glioblastoma multiforme, we found that we could recapitulate the SIM group, which was identified earlier by the TCGA, and that we could recapitulate this group exactly, and this with a smaller number of genes. And we also characterized the significant hypermethylation that is characterized by this subgroup. Next, we also found a small subgroup, a new subgroup in ovarian cancer. And this subgroup was characterized by 54 hypomethylated genes and about 140 hypermethylated genes. And these were genes that were enriched in differentiating T lymphocytes expression programs. We also found that these patients had higher CD4 and CD25 expression. So this was indicative of patients that have high infiltration of T cells that are characterized by these 2 markers. And this is known to promote tumor progression in the literature, and we could also see that these patients had a poorer survival. Next, using these DM values, we also built multivariate survival models by using a Cox survival model with a lasso penalty. And we built these models using different set of genes to start from. So we used all genes, at both the methylation or the expression level. We used only differential genes, only functional genes, or the metal mix genes. And what we found was that when we used the metal mix genes based on their DM value, we had the best prediction of survival. If we compare that, for example, with the 10% most varying genes when using just gene expression data, we also compare this with using the 10% most varying genes based on just the raw methylation data. And we also looked at the 10% most varying differential genes at both the methylation level and at the DM level. And only when we focused on both functional and differential genes, we found the best results predictive of survival. Finally, we also focused on one other gene, which is called MMP7 in breast cancer. This gene is significantly hypomethylated in basal breast cancer patients, and hypermethylated in luminal breast cancer patients. We looked in breast cancer cell line methylation data, and we found that this was also true, that in basal cell lines this gene was hypomethylated, whereas in luminal cell lines this was hypermethylated. Similarly, at the expression level, we found that for basal cell lines, we found a number of cell lines that had high expression of this gene. And we selected two cell lines, HCC1143 and HCC70, that had high expression of MMP7. We designed small interfering RNAs, and we were able to knock down this gene significantly. And then we found that when this gene was knocked down, that cell growth was significantly inhibited. So to summarize, we developed an algorithm called MethylMix to identify hyper and hypomethylated DNA methylation. We also incorporated normal methylation data and paired gene expression data. We investigated the genomic location of these CpG sites. And also we defined the DM value, which is the differential methylation value, and showed this value to cluster patients and also to predict survival. And finally, we focused on one gene in breast cancer and validated this gene. So I would like to thank you for your attention, and we'll be happy to answer any questions.
Time for one question.
Thanks for the talk. I'm just wondering, when you're talking about methylated genes, are you talking about just the promoter region or upstream region? Did you look at the exons?
So this platform is mostly profiling the promoter region of these genes. So these are only 27,000 CpG sites that are profiled. So this is mostly, and these CpG sites are mostly in the promoter or in the gene body.
Okay.